at-home microbiome testing is very important. I think that today we have many options as citizens, if we are curious, to obtain the picture of our microbiome. So it's like the selfie of the inside. And I think that it's fair to be curious. When it became possible to have your sequence of your genome, then people were curious and they wanted to know what their genome looked like. And so they would pay for it. And today you can pay to have your microbiome sequenced. But the way I view it today is that there is no medical use for those at-home tests that are proposed for the moment, essentially because it's not standardized. So what do I mean by that? I mean by that if you were to send aliquots of one stool sample in 3 different laboratories, you would get essentially 3 different pictures because the process is not exactly the same. And you can imagine that if you provide the result of such a test to a medical doctor, then it's really complicated to manage that because you will know that if it was another lab, it would be a different picture. And what can you tell from that? So the idea as of today is that for me, it's fine for curiosity. It's probably also fine for wellness if you use always the same laboratory to obtain the result, because then you can compare from one time point to another. And so it makes sense. What you want to modify, then you can have the results of the next time point and know whether you have been effective. But if you think of medical doctors using the information, then we're not ready yet. So then what do we need? We need the standards. And we already published on the standards, so they are available. So you can imagine that a medical biology lab will be able to apply standards that will be so accurate that a lab in the US, in Europe, in Australia will give the same results. So that's fine. You need big numbers because you want to compare your results to a large reference of information. And so in that respect, we are making progress every day. I'm scientific coordinating a program in France where we are recruiting 100,000 volunteers to give us stool sample to analyze their microbiota. And this is part of the Million Microbiome from Humans project. So it's talking of 1 million microbiomes being characterized today worldwide. And that will give obviously much more information than we do have today in terms of reference. And then you're not there yet. You need to demonstrate that the use of the microbiome test will come with clinical benefits. It's really setting yourself into the evidence-based medicine, basically. So you want to set up a trial that will allow to demonstrate that if the medical doctor uses the information on the microbiome, then clinical management is actually better. And the outcome for the patient is better than if you don't take it. And there are a few lines of example that are really reassuring in that respect. And I can cite a few of them. For example, we were able to show that just microbial diversity or richness in terms of distribution is bimodal. Bimodal means you have a population with low richness and a population with high richness within healthy individuals, around 5 to 10% of low richness, possibly more at threat of chronic conditions. When we looked at that for moderately obese individuals, it was up to 40% of the population. And we were able to show that it was predictive of non-response to a low-calorie diet. So those individuals, yes, with obesity, moderate obesity, and low richness of the microbiota, we were not able to lead them to lose weight, but also to improve their inflammatory status and to improve their diabetes parameters if they were moderately diabetic. And more than that, we looked at that for liver disease patients. And what we know is that low richness is associated with the severity of the condition. So as you move from steatosis to NASH to cirrhosis, then the more severe you go, the less or the higher the fraction of the low richness within the patient population. And in cancer and cancer treatment, it's also very informative. It's in immunotherapy, which is really the most modern treatment of cancer today for lung cancer, for example. We know that 50% of the patients will not respond. And the testing of the microbiota, especially with the parameter of richness, will predict whether the patient will be a responder or non-responder. Wow. Which means a lot for the clinician. When you know that it's 50% that will not respond, and some cancers it's even less than 50% responders, then you want to know, and the next stage you want to know what you can do to help those patients to respond.